So hi, this is part B of the hypersensitivity lecture. And what we're going to talk about in the second part of this lecture is the other three types of hypersensitivity reactions. So I'm going to talk about the mechanisms and provide some examples for type 2, type 3, and type 4 hypersensitivity reactions. So if we start with type 2, so this can involve two mechanisms, and it's basically the immune system responding to an antigenic cell. So what I mean by this is a cell that is expressing antigens that the immune system is recognizing as foreign particles. And generally speaking, this will arise from a loss of tolerance. So we talked about the importance of central and peripheral tolerance. If those mechanisms are bypassed for whatever reason within a given individual, then type 2 hypersensitivity reactions may arise. And this is where the immune system, whether it be cells or complement, will target a host cell that is expressing a host antigen that the immune system, and specifically antibodies and complement, are not recognizing as being a host protein or peptide. So there's two mechanisms, as I've said. So the first mechanism is shown here. So this is known as an antibody-dependent cell-mediated cytotoxicity mechanism. So we touched upon this very briefly in one of the past few sessions that I gave you. And this involves a cytotoxic T cell or a natural killer cell. So those are two good examples. These cells will recognize a target cell. So they'll recognize the surface antigen on a target cell. And because they are recognizing this, they will then basically have some sort of effector function. And because they're cytotoxic cells, they'll produce things like enzymes that can drive the degradation of the target cell. The other mechanism that can also be involved in this process is the formation of a membrane attack complex via activation of the complement cascade. And what will happen here is you will have antibodies recognizing the surface antigens of a target cell, and this recognition will basically drive activation of the complement cascade. And this is highlighted quite nicely in the YouTube video from about 10 seconds onward. And two good examples of type 2 hypersensitivity reactions are transfusion reactions and also an autoimmune disease known as hemolytic anemia. And these are highlighted here. So you have in the issues with the blood transfusions, this can basically drive this type of hypersensitivity reaction when you have a mismatching of blood types. So of course, this very, very rarely happens, but it's not unknown to have happened in the past. Basically, what happens here is you have a donor that has red blood cells that express type A antigen being given to a recipient that has type B blood cells. So basically, what will happen there is that the antibodies will not recognize the type A antigen on the red blood cells, and therefore they will basically try and attack those red blood cells and drive the formation of or drive the process of the complement cascade, and also drive degradation of the red blood cells. The autoimmune disease, as an example, known as hemolytic anemia, can arise when the host body, the immune system, cannot recognize its own red blood cells. And because it can't recognize its own red blood cells, it will drive an inflammatory response against them. And this inflammatory response, in a very similar manner to here, will involve the activation of cytotoxic T cells or natural killer cells, or the complement cascade. That basically the primary aim of the immune system is to drive destruction and lysis of these red blood cells. And this can arise in patients with hemolytic anemia. In terms of dental related examples, there's very limited examples that I'm able to provide to you, but you just need to be aware of the process of type 2 hypersensitivity reactions, and these two examples are probably the best ones to go away and revise for your exams. Type 3 hypersensitivity reactions, as I've said, this involves the formation of antibody-antigen immune complexes that can be either found systemically or deposited within tissues or organs. And this is immune complexes forming, these are antibodies forming these complexes against soluble antigens that are found within circulation. And presence of these complexes will drive different processes, such as activating the complement cascade, recruiting neutrophils to the site of the complex, and also other inflammation-mediated damage. Again, the YouTube video, very good. A very good schematic is shown in the YouTube video from about 10 seconds onwards for this process. Example-wise, there's 3 examples here. So you have a disease known as Arthur's reaction. You have serum sickness, and within the context of the oral cavity, you have oral erythema multiforme. So if we just talk about the examples in a bit more detail, so I've included Arthur's reaction as an example because I think it's quite fitting given this part here. So this is a localized hypersensitivity reaction. It's basically localized within blood vessel walls of the found within certain tissues. And as I've said, it's quite fitting because it can arise from injected particles such as booster vaccinations. So of course, given the current climate with COVID and the vaccines being rolled out at the moment, this is a good example to think about the process of type 3 hypersensitivity reactions, because this mechanism here, highlighted in this figure, will arise within certain individuals. So basically what happens is you have injection of the antigen into an individual. And basically what happens is you get formation of these complexes within the tissue. So the antibodies will recognize the antigens within the tissue and they'll drive an inflammatory response to them. And this inflammatory response can lead to local inflammation, increased fluid and protein release into the tissue, and also other processes that can ultimately lead to swelling of your tissue. And of course, this is in certain individuals is what you will find when you have the booster vaccination, is that basically the deposition of these complexes within the tissue of your skin where you have the vaccination will basically sorry, I think I got cut off there. Apologies. So what I was saying was you'll get local inflammation, increased fluid and protein release into the tissue. And of course, this is why you would get reddening of the tissue after, and possibly pain within the tissue after you get the vaccination. And that is predominantly driven by this type 3 hypersensitivity reaction within the skin tissue. There's other examples. So serum sickness is ultimately a systemic form of Arthur's, where you have these complexes, these antibody-antigen complexes circulating and lodging in different tissues. But the mechanism is very similar to as we described in the previous slide. In terms of the oral cavity, you have this oral erythema multiforme, which is characterized by blistering of the oral mucosa. And it's predominantly because of deposition of these immune complexes, specifically IgM-bound immune complexes within the vasculature of the oral mucosal membrane. We don't really know the reason behind this disease. There is some suggestion in the literature that it's an acute inflammatory response to certain viral infections, such as herpes simplex virus, or it can be driven by hypersensitivity to certain drugs. But we don't really know exactly what the reason is behind this disease or the pathogenesis of this disease. But this is shown here on the right-hand side, and this is a type of an example of type 3 hypersensitivity reaction. So if we move on to the final type, type 4, so this is ultimately a T cell mediated hypersensitivity reaction, so it doesn't involve any antibodies. It sometimes can be called delayed hypersensitivity because Because of the process, it involves recruitment of T cells. And as you should know by now, T cells are part of the adaptive immune response, which can take up to a few days to actually kick in and function within the immune system. Ultimately, what happens is you have a localized T cell reaction at the site of the antigen or the allergen exposure. For example, within the skin, contact dermatitis is a good example of a type 4 hypersensitivity reaction. And it can involve not only T helper cells, but also cytotoxic T cells as well. And basically what will happen is a T helper cell will produce cytokines, and these cytokines will instruct cytotoxic T cells and macrophages that are resident within the tissue to basically drive inflammation. So as I've said, contact dermatitis is a good example. The rejection of certain tissue grafts is another example. Within the context of the oral cavity, there is some suggestion in the literature that OFG, which we've talked about in the chronic inflammation lecture, may actually be an example of a type 4 hypersensitivity reaction, where basically you have T helper cells and also cytotoxic T cells basically interacting with the tissue resident macrophages. And in this disease specifically, you'll get this formation of the granuloma within the tissue. And we don't know exactly why the actual mechanisms behind this disease, but there is some suggestion that it's a hypersensitivity reaction to some allergen within certain patients. This is a final recap. I think we've flown through the second part of the session, which is good. So you should be aware after today's session that there's 4 main types of hypersensitivity. These are classified using the gel and Coombs classification system. Type 1 is an IgE antibody mediated response. There's a prominent role for mast cells and basophils and antibodies, specifically IgE in this response, and ultimately it's the immune system responding to a certain allergen, or in certain cases, a hapten that is conjugated to a protein. Type 2 involves IgE or IgM mediated responses, leading to either activation of the complement cascade and or a antibody dependent cell mediated cytotoxicity reaction. So basically what happens here is that you get activation of complement, which can lead to the formation of the membrane attack complex, or you can have activation of cytotoxic T cells or natural killer cells, which can drive destruction of the target cell. Type 3 is an example of antibody-antigen complex formation within either systemically, so within the circulatory system, or these complexes can be deposited within certain tissues or organs. And then finally, we have type 4, which is T cell mediated. So no antibodies are involved in this process. And there is some suggestion that OFG is potentially an example of a type 4 hypersensitivity reaction. So I think that more or less concludes my session. So that's all 10 lectures complete. I do appreciate that the content that I've given you might be quite challenging for most of you. Hopefully I've simplified it enough. As I've said many times, if anyone has any questions, any queries, concerns about the content, especially when it comes to revision, please don't hesitate to get in touch with me. You all have my email address. I think there should be another tutorial. Tutorial 3 should be including some of the last few lectures that I've given you. And what I've got in this next slide is just some revision tips, which I hope it may not apply to all of you, but some of you may find this of use. So what I would suggest is to group my content of lectures into the following sections. So if you think about the first line of defense against a given infection, for example, then of course you need some branch, or you need the involvement of the innate immune system. basically kick in as the first line of defense. So when we're talking about the innate immune system, then of course acute inflammation is probably the best place to start. So I would group the acute inflammation lecture 7 with the innate immunity lectures 3 and 4. And generally speaking, in acute inflammation, hopefully you should know by now, acute inflammation is generally always followed by regeneration of the tissue. So you get resolution of disease, and we've covered that in lecture 9. So that would be the first thing I would worry about learning. And this should give you an idea of how the immune system responds straight away to a certain disease or in a certain disease. The second thing I would then group together is the chronic inflammation lectures and also the adaptive immunity lectures. So lectures 8, 5, and 6, as obviously there is a prominent role for the adaptive immune system and the adaptive immune response within the process of chronic inflammation. So if you can recall, chronic inflammation is this persistent bouts of inflammation within a given tissue. And because it is persistent bouts and it can occur weeks, months, even years, then of course you have some level of adaptive immunity playing a role within the inflammatory response. And then because it's chronic inflammation, generally speaking, you get tissue regeneration and repair, and you don't really, you see the formation of scars within chronic inflammatory diseases, and we've covered that in lecture 9. So the second part of your revision should be predominantly looking at chronic inflammation, and I think the adaptive immunity lectures kind of go hand in hand with chronic inflammation. The first 2 lectures that I gave you generally encompass all the above content. So obviously, when you think about pathology of disease, basically it's all the processes that arise during the disease. And of course, this is covered by acute inflammation or chronic inflammation, depending on the disease. Defense cells will be, of course, talked about in each of the individual sessions. And then finally, we have lecture 10, which it's a unique lecture in that it more or less involves most of the above topics to some degree. It is a bit of a standalone lecture, and unfortunately, you need to at least have some understanding of hypersensitivity reactions. But as in terms of revising this, I would worry about acute inflammation. So basically what's happening first in the immune system. Chronic inflammation, what happens if you have persistent bouts of inflammation? So your adaptive immune system is really important to consider here. And then finally, hypersensitivity is just a standalone lecture that you need to go away and revise. But that brings us to an end for my sessions. As I've said, please don't hesitate to get in touch with me, and best of luck when it comes to your exams. So thank you for your attention, and I will see you hopefully in BDS 2 and possibly BDS 3 when I may have to give some lectures to you. So thank you again.